Hello, my name is Ruben Escobar. I'm enrolled in the Mammalian Physiology Section 8 class, and this is my journal article presentation. My article is titled Variation in Heart Rate During Static Breath Holds in Bottlenose Dolphins, and this article was authored by the following individuals: Andreas Fallman, Bruno Cozy, Mercy Manley, Sandra Jabas, Merrick Malik, Ashley Blawas, and Vincent Janik. And this study was done in 2020 at the location of Roy's Dolphin Habitat in Las Vegas, Nevada. This article was found in the Journal of Frontiers in Physiology. A little bit of background in physiology on the study. This study was done on bottlenose dolphins, and dolphins are one of the groups of mammals that use a dive response to survive underwater, essentially meaning the amount of time that they're able to hold their breath to be under the water and do the necessary things they need to do to survive. When this happens, they enter a state known as bradycardia. Um, Dolphins aren't the only mammals that can do this. Other marine life can do this as well. We could also see this in humans, primarily athletes, since their hearts are conditioned to have a lower beat per minute. And we can also see this in older individuals as well, just because the electric activity in the heart slows down with age. So yeah, bradycardia, we can see is the slowed heart rate during breath holding, which essentially allows them to hold their breath for longer periods of time. So this study also wanted to see if this breath holding conserves oxygen and reduces the energy use. It could also limit nitrogen absorption during the dives because too much nitrogen in the body can cause problems, especially diving for long periods of time. And the researchers thought that the automatic response for the bradycardia, as far as like holding their breath for long periods of time, was something that happens automatically, that is not trained or is not learned. But the new research wants to test to see if it's something that's learned and it's adaptable, if it's something that the dolphins learn how to do over time. So, research question and hypothesis. The main research question that these scientists wanted to figure out was if the dolphins can adjust their heart rate based on expected dive durations, um, based on cues that are given, if the dolphins are able to hold their breath for longer periods of time, and how they can manage the BPMs. Hypothesis for this would be a dive heart rate is something that's conditioned and it's not purely reflexive, meaning that a heart rate is something that can be trained to last for a longer period of time, and it's not something that you're born with, like how uh, other mammals and reptiles have a biological clock as far as knowing what to do when they're born, um, and it's not a learned um, situation. So the researchers tested whether the dolphins could learn and modify physiological responses, and they wanted to compare long dives versus short dives, and also a no signal dives. So this study was broken up with the cues given to the dolphins in three categories. Long dives, cues were given for the test to tell the dolphins to dive for long periods of time, one for a shorter duration and one with a no signal at all. So the dolphins didn't necessarily know what type of dive they were going to be doing. And they wanted to see if the anticipation with those cues affected the heart rate changes within the dolphins. So the goal ultimately was to determine if the dive response was something that is learned or if it's something that happens automatically. The methods and summary. So how they did the experiment. So the experiment used three bottlenose dolphins that were at the Nevada location, um, and these dolphins were selected at random. So the experimental design wanted to use visual cues, as stated in the previous slide, to account for dive durations. So split up into three different types of dives. A longer dive, which is between 120 to 180 seconds range. A short dive, which was about 30 seconds, and a dive where no cues were given at all. So there was no signs indicating that how long the dive was going to be for the dolphins. So it was a randomized trial that took place just to eliminate any bias and variability between the dolphins and the trials. And this was monitored through the electrocardiogram to monitor the heart rate during the dives between the dolphins. And they wanted to compare this across the different dive durations to see if the electrocardiogram would show a pattern or if it would show more of like a um, random situations. And so this design allowed the testing of the dolphins to predict and adjust the heart rate based on the cues that were given. So this is one of the figures that were inside of the article. And this figure is basically showing the dolphins that the dolphins lowered their heart rate during the diving, and that the amount and the how quickly they dropped their heart rate depended on whether they expected a long dive, a short dive, or a dive where they had no control over. Basically, it was like a surprise on how far they'd be diving and how long. And so figure two in the article, it shows the individual dolphin statistics with the tests, and it highlights that the heart rate control is a lot more consistent when they are giving cues. So this meaning that the dolphins did show a pattern when given certain cues than not. And when they weren't given any cues, the heart rate for these dolphins were a little bit more unpredictable. The discussion and conclusions. So the heart rate changes depending on the expected dive duration. So based on the study, it was shown that the dolphins did have a change in heart rate depending on how they saw the cues that were given to them before their dives. So they were able to adjust their heart rate to adequately suffice each trial. So for the longer cue dives, they saw a faster and deeper bradycardia. So they were able to lower their heart rate even more to account for those long dives. Dives that were seen with no cues or no signal for the dolphins showed a more variable heart rate response. So it was more of a um, random pattern, you know, rather than a test with the cues with the long dive that showed a um, a conditioned heart rate. And so the test ultimately showed evidence of a conditioned physiological response, as I previously stated. And the dolphins are able to predict their dive length and adjust their heart rate based on that, on the cues that were given. So ultimately indicating that the dive response in these dolphins is something that is learned and can be accounted for when they are given tasks like this, and it's not just flexible, and it's not always the same every time they dive. So I would say, so for the critical analysis and the future directions, how I see it is a weakness that I saw in the study would be a small sample size of only these three of the three dolphins that were tested, because it limits the amount of the true um, population size and these dolphins that were raised in captivity, they could all just be trained to those conditions, which gave those results. Um, as I said, it was conducted in a lab, so it wasn't their fully natural environments like how they are in the wild. So that might be a factor in the results that were shown. And there aren't, it's not fully reflective of the wild behavior out in the ocean because there's certain factors that don't play into it as far as other food options that are on the area. Maybe there's predators. So that could be a reason why we saw these results as well. So I would say that would be something that could be improved on for this study. And a future scientific question that I have would be to follow the um, same question, but to study the wild dolphin population rather than three dolphins that are raised in captivity and that are in a lab environment. I think that the dolphin populations out in the wild would show a more accurate representation of how they manage their heart rate, just due to all the factors being put into place in the wild rather than in a lab. And increase the sample size because I think three dolphins isn't a big enough size to get a accurate representation, even though the three showed you know um, good results in terms of the cues that were being given. I think that a larger sample size would be even better. And then obviously to compare the results with that study to this study and the dolphins that were tested in the lab. All right, and then this is the reference page. I used the journal article that I found using the CSULB library from the Frontiers in Physiology journal. Thank you.